Pleasant morning to you all, children. We have already discussed the digestion process. How does it takes place in the mouth, stomach, and small intestine. Now, this is just the summary, just a quick recap. Digestion in the mouth, as I said, it takes place, that is called as mastication or mechanical digestion, which involves the breakdown or cutting of food into small pieces with the help of the teeth and mixing of the food with the help of the tongue. And chemical digestion involves the salivary amylase present in the digestive juice secreted from the salivary glands. Salivary amylase, also known as ptyalin, it acts upon the substrate. Substrate means the nutrient. So this ptyalin or salivary amylase act on the carbohydrate part of the food, that is the polysaccharide, and breaks down into disaccharide. Clear? So in the mouth, what happens? Polysaccharides are broken down into disaccharide, and that is facilitated by the digestive juice, saliva secreted by the 3 pairs of salivary glands. Then we have the next part, esophagus. We have also mentioned it earlier that it is this particular organ do not have any digestive juice. They just have the function of peristalsis, that is pushing the food down from the mouth to the stomach, and that wave of constriction and relaxation of its muscular walls pushes the food, which is in the form of bolus, into the stomach. In the stomach, the food, the gastric gland secretes the gastric juice, which also contains concentrated hydrochloric acid. The enzymes present in the gastric juice are pepsin, which is in the form of inactive form, pepsinogen, which is converted into active form by hydrochloric acid. They work or act on proteins and break down into its product, that is polypeptides, proteases and peptones. While renin, it acts on milk protein and form casein protein, that is involved in curdling of milk. Then we have the small intestine, which is grouped or divided into duodenum, jejunum and ileum, whereas the digestion part takes place in the duodenum and ileum. Absorption part of the digested food takes place, whereas jejunum act as a link between duodenum and ileum. Duodenum, the upper part of the small intestine, as you have studied earlier, it receives the 3 digestive juice. One is bile juice from liver, pancreatic juice from pancreas, and intestinal juice from intestine. What is the role of bile juice? It is stored in the gallbladder and it acts upon fats. Bile juice also do not contain any enzyme, but it contains bile salts and bile pigments. It acts upon fats and breaks down into smaller globules, which is referred as emulsification of fats. It also makes the medium alkaline. In the stomach, the medium is acidic, whereas in the mouth, the medium, the pH, is also alkaline. Whereas the pancreatic juice secreted from the pancreas, which is a white leaf-like structure organ, that is the gland present just below the stomach. It contains amylopsin or pancreatic amylase, acts on starch, any leftover starch, and breaks it down into maltose. Trypsin, it again acts on the leftover proteins which are not digested in the stomach into polypeptides and finally break down into dipeptides, or it is end product amino acids. Intestinal juice, it contains the enzymes erepsin, peptidase, which acts on polypeptides and dipeptides and finally break down into its last product, amino acids. Whereas maltose are acted upon by maltase present in the intestinal juice. Intestinal juice, other name is succus entericus. It acts on maltose and forms the end product glucose, while the enzyme lactase acts on lactose and forms glucose and galactose. These are all the monosaccharides. Then when we talk about the large intestine, we have the caecum, colon, ascending, transverse and descending, and rectum. Colon, no digestion takes place. Here what happens, the undigested part of the food, it is transported to the rectum, and while its movement through the colon, the water from the waste material or the waste products are reabsorbed so that the waste product becomes solid form, and where it is temporarily stored in the rectum and finally adjusted out. After digestion, the next step is assimilation. That is what happens to the end product of digestion of starch, proteins and fats. Now, when we talk about the carbohydrates, that the glucose, vitamins, minerals, amino acids, they are absorbed in the ileum part by the blood and transported to different parts of the body. But what about fats? They are not absorbed by the blood directly. They are absorbed by lacteals, by the lymph vessels, and finally they enter the blood circulatory system through the venous blood. Now when we talk about, there are certain tests that you should know. I'll show you in the laboratory how to identify that whether the given food sample is a carbohydrate, whether it is a protein or a fat. First of all, let us discuss about the test for glucose. For that, what do? We take some amount of the sample, the glucose solution or the food sample, and add Benedict's solution, equal amount. Got it? Now, when you heat it, when we heat the food sample, what we have taken? We have taken small amount of glucose and added some amount of water to it, made a solution and added Benedict's solution. Clear? Now, when it is heated, if glucose is there in the food sample, I have told you it is glucose, but if unknown sample is given and you are asked to test for glucose, what do? You take that unknown food sample solution, add Benedict's solution to it and heat it. What do you get? A brick red precipitate. If a brick red precipitate is formed, that means the given food sample contains carbohydrate. Another test for carbohydrate, that is glucose or sugar, is that instead of Benedict's solution, you take Filing solution. Same way, if you get brick red precipitate, it is the given food sample contains sugar or glucose. Got it? Now for the proteins, what do? I'll show you in the laboratory. This is not there in your syllabus, but you should know it since it is there in your practical. What are the chemical used? You have in the protein test sodium hydroxide and nitric acid. Got it? And the food sample. So when you add, first you add nitric acid, then you add ammonium hydroxide, you get white precipitate, and after that white precipitate, after you add ammonium hydroxide, it turns to yellowish orange precipitate. Clear? So that is all. Have a nice day. Stay home and be safe.